Our next speaker is Brandon Munn from the University of Sydney, in my lab. Hey, Brandon. He's going to be speaking to us today on behalf of Gabriel Wainstein, who unfortunately couldn't make it to the meeting. And he'll be speaking about catecholamine-driven low-dimensional brain dynamics and cognitive task performance.
Great. Hello, OHBM. Thank you very much for all hanging around. My stomach is hurting, so I'm sure all yours are. Thank you. All right, so I can just click this button. Cool. Great. Lovely. Cool. All right, yes, I am, we're moving around the place. Yes, I am Brandon, as Mac mentioned, and I'm presenting here for Gabriel, and I'm going to just try and move this slide. There we go. So here we go. Here's Gabriel, the man. He was a PhD student from our lab. Now he's a graduated doctor, Dr. Wainstein, and he is a great little scientist, and I'm going to present some work that we worked on together, particularly the role of the ascending arousal system. Our lab is particularly interested in the subcortical subcortex and the ascending arousal system, the thalamus, and how that can constrain cortical dynamics. So, but particularly today, I'm going to be talking to you about the locus ceruleus and how noradrenaline can change brain dynamics. So the reason it's really interesting, brainstem pontine structure, very few tiny cells, but these few cells have a huge effect on the brain. They project really diffusely across the brain, if you can see on the left here. And these few little cells are really important because if we think about, so they're really important, like fight or flight dynamics, any kind of extreme moment, your LC is going to be changing its activity. And what happens is, if you think about your brain, relatively stable structural on the timescale of seconds, no plasticity happening on that stage, but from the change of noradrenaline neuromodulatory change on your brain, your whole behavioral cognitive function can completely change. So at low levels of noradrenaline, you can be really drowsy and sleepy, and in the same next couple of seconds, like right now when you're on a stage, you can have high tonic levels and just start freaking out. Or in the middle zone, you can have this inverted U, optimal performance at intermediate noradrenaline levels. So, there's this crazy situation where you're having a static fixed brain, but just changing this subtle neuromodulatory level, you get completely different cognitive functions. And the question is how are these different cognitive capabilities emerging from just changes in the brain dynamics and not the actual physical brain? So, the way I'm going to unpack this is I'm going to talk about, first, is a way I think my background is physics, so I'm really interested in brain dynamics and how dynamics change. So that is how you get one state, if you have a state of X of t, how that brain state and how it changes from 1 second to the next. And so there's two main ways to look at brain states. You can think about probably the more common view is what I would call an allocentric or a bird's eye view. If you think about your little bird flying over this little map of the world, you can sort of look at each of your little brain states, maybe that yellow house is your resting state, one of your resting state networks, the default mode over here, then there's a tension network over here, and so maybe Markov transitions is another example of the allocentric view, and they're talking about absolute brain states, activation patterns. But another way you can look at brain is looking at dynamics from an egocentric point of view. So there's the first point of view. This is a relative framework. It's kind of looking at derivatives versus absolute values. And you can think about it as so your first person view is as if you're driving a car now around this little brain map, and you can asking how does my brain dynamics change relative to a given position. This will make sense later, but I think the point I want to start and say is it's sort of really important to think about how different brain frames or reference frames and how if you look at brain activity in different ways, those different ways I think are going to give you different insights into the function of the brain. And if you find the right frame or the way to look at your brain activity, a lot of the behavioral effects that pop out I think will make a lot more sense. So that being said, I'm going to now talk to you about a little technique that we developed in the lab. So this is we've really borrowed the idea of some physics techniques. This is from actually using a little reframing of the Boltzmann equation, which is fine to do. Claude Shannon did it. He created the information theory. So hopefully we can make some progress using that approach. But the idea is, if you think about the brain and brain dynamics as existing on some sort of low-dimensional manifold, the metaphor here is you think about a ball rolling around a little sheet. Perhaps there's evidence of the brain being low-dimensional, so it's not just a metaphor. It's actually an example saying if you think about this ball, which is your current brain state, it might roll around this lower-dimensional landscape. And some of those states, right, are going to be like wells over here where this ball moves around. Say your state 1 here. So again, we're using like a bird's eye view, right? We're looking at this like low-dimensional state, and this ball, like this, might move around and might go up up to a little high hill and fall back down. So now, if you think about these states or where the ball like resides a lot, they're going to be really so the ball will always fall to a low energy minimum. So that's kind of like thinking about relating probability to energy. So it's inverse probability. This is the Boltzmann equation here. And the idea is, if you think about like the old chemistry diagrams over here of reaction pathways, where you can have your like sort of state 1 over one side, and then some sort of energy barrier, and then you trying to push your ball or the state into this second state 2. We can reframe that exactly in that dynamical perspective into looking at brain dynamics. Cool. So we first went, okay, that's a really nice idea, but like, what do we even think that is going to do in the brain? Well, from like some previous work, we theorized that perhaps if you imagine your like a low-dimensional brain state, we thought the locus coeruleus, so your cortical noradrenaline, might flatten that state, so suddenly makes it easier for your brain to jump from one state to another. And then this is just also, we looked at the opposite, so like the basal nucleus are inert, so like the cortical source of acetylcholine. And we thought actually this might do the opposite. It might deepen some of those wells, so make states more stable. So if we think about something about locus coeruleus making things your brain dynamics more flexible, the other one making it more stable, we were wondering if we could see any evidence of that in the actual brain dynamics. So if you want to think about it in the sort of like this reaction pathway state, we hypothesized that the locus coeruleus might lower that energy barrier, and the acetylcholine might increase the barrier, so making it harder to do state transitions. And here's where I took this idea of this egocentric view. Now we reason that like the egocentric view might be a better fit for neuromodulation or neuromodulators, because they're sort of evolutionarily so ancient that it was less about like controlling a brain perhaps, but more about changing like yourself in an instant. Say I'm giving this talk, it doesn't really matter what situation I'm in. If a tiger runs into a room right near me, like I need to quickly change my brain states. It's a relative, neuromodulators are very like relative and like sort of reference frames of where you are right now. It doesn't really matter. You could be on a really, you could be doing a test, or you could be in like a really important moment of your life, and you're trying to like your acetylcholine might be trying to keep you maintained and like keep you coherent in what you're doing. So that we reason this reference frame, the egocentric, would be useful. And I'm going to sort of unpack the way to look at it in this bottom here. This is from baseline activity. So I've got my 70 fMRI. I've got time series of my locus coeruleus and basal nucleus inert. And then I've looked at like periods where following activation of those, so like high bold activation of those two regions. And then I've looked at other periods outside of those windows. And this is like my general landscape from my egocentric view. And if you think about it, so if you think about putting a little car on this, you can see my mouse, this little corner here, and you're looking out, so you've got your time and then your mean square displacement. So that's like, so time is sooner or later. So this is like what's my landscape or my likelihood of changing my brain state soon or later, and am I going to change that brain state by a little or a lot, like small amount or a large amount? And then the energy is related to my like how surprising or is expected that brain state change. So if I'm standing in this little position here, I look to my left, it's really unlikely that I'm going to change my brain state by a large amount. So it's much more likely I'm going to change it by a little. And then after some sort of given 10 TR, there's like some sort of generic amount of likelihood of being in this brain state here. But the real question is, the real insight is when you can now repeat this for, as I said, of those periods after large activations of those two neuromodulatory centers, and I can repeat so calculating probability distributions and calculate the energy of those transitions, and I find the locus coeruleus really flattens my brain state, whereas the basal nucleus inert increases it. So we see actually doing this approach, we get a direct sort of like test and validation of like this hypothesis of how neuromodulators may change your brain state dynamics. So we've since repeated that approach in lots of different things. We've done sort of like behavioral, looking at repeated in different data sets, looked at structural connectivity. We've done it in models and large-scale models, micro-scale models. We've shown some causal validation in rats and now primates for acetylcholine and noradrenaline, respectively. But the next one we wondered was really like, have we got any causal, sort of, at least semi-causal evidence of this in humans? So Gabriel looked at some humans, where looked at particularly like the COMT, which is a catechol, like regulator, so it like degrades the catecholamines in the brain. And there's also a genetic this single nucleotide polymorphism, which basically can modulate how quickly the brain can degrade noradrenaline. And long story short, so there's a big data set that was previously DNA tested. We got some DNA samples from the people, tested them for their pharmacology amounts, and we could tell whether they've got whether they degrade noradrenaline quickly or slower. And long story short, so now we're looking at these same energy landscapes, so we take relative ones, and we can actually see a precise prediction, right? For those who degrade their noradrenaline slower, the energy landscape is deepened, right? So like we're taking the difference here. So this was sort of like a rough causal demonstration that what we predicted is case, at least causally in humans as well. So to summarize, we think dynamics matter in the lab, and they can drive functional insight into how the brain is working. You want to really choose the right reference frame to get that sort of insight. When we did it by using this egocentric view, it really was universal and consistent across species, the scales, and whatever you want else. The locus coeruleus, as I said, very conserved, and there are some genetic differences that seem to align with the flexibility. And this was nice because people have looked at those that COMT regulation and sort of related to cognitive flexibility. So now we've got sort of a mirror and match between cognitive flexibility and actual brain dynamic flexibility. And finally, all of this code is available on my GitHub and also the lab's GitHub, so it's very easy to use. Feel free to reach out to me or anyone in the lab. And then finally, there's our great little Shine lab. So we've got actually my post is on in Wednesday and Thursday. Come see mine at the same time as mine. Near us is Bella, and Annie's is also on the same day. And then Josh, come visit his poster tomorrow. Thank you.
All right, we have a question from the app. It says, great talk. Sounds like an egocentric view maps nicely to fast neuromodulatory changes. So does it follow that an allocentric view would map to slower timescale neuromod changes, so over minutes or more?
Ooh, that's a good question. I actually I'm a big fan of the egocentric view over the fast timescales. I think that makes sense, for fast fluctuations and more immediate sort of actions and responses. But for the slower timescales, I think a lot of, if you would believe, if you look at fMRI, a lot of the slower timescales, should I say, really are where you get that strong explanation by low dimensionality. So your resting state patterns, and you see these really a low dimensional framework for mapping these. So when we're saying low dimensional, or a lot of patterns explain the brain activity, that's really aligning with an allocentric view. So yeah, I think the slower dynamics would be really aligning with an allocentric view and should be better explained. But I will say, a cool way of using this approach, right, is you can combine the two as well. So you can start looking at egocentric and allocentric across the brain. And I think there's going to be different parts of the brain that use different sort of reference frames in a way, right? Probably something like the hippocampus, the visual field, are probably going to be more aligned to an allocentric view, where there's clear and immediate meaningful locations that are representing some sort of clear pattern, and then maybe things the brainstem, or maybe different parts of the hippocampus, the entorhinal cortex, maybe, and maybe frontal would be more aligned to sort of an egocentric view.
All right, thanks very much.